Hi everyone, I'm Suanna Moran,
And I'm Elizabeth Bolton. We're both seniors majoring in computational neuroscience.
and I also have an additional major in psychology. Today we're presenting on research investigating cognitive control development through behavioral, qualitative, and neuroimaging methodologies.
I'm going to start by giving a quick overview of what we are going to be talking about. Our presentation is going to cover two studies. Our first study was a validation study done with adults. It validates an fMRI resting state measure using fNIRS with 18 to 21-year-olds. We collected that data in the spring of 2019.
We're also going to talk about a longitudinal study that included 4 to 5-year-olds, where we looked at how cognitive control and associated neural substrates changed over time. We first pre-registered our longitudinal study in the fall of 2018. We started pilot testing and then collecting our longitudinal data in cohorts of children. We had to abruptly stop data collection in the spring of 2020 due to the current pandemic. But let's first dive into explaining some terms, starting with cognitive control.
Cognitive control is a set of processes that support adaptive, goal-directed behavior in the face of changing task demands. For example, in a school setting, this would be paying attention to a class lesson and not getting distracted by classmates. This ability is important because cognitive control in children has been shown to predict many factors when they're adults, including academic achievement, likelihood of committing crimes, and occupational success. Furthermore, several developmental studies provide evidence that show significant cognitive control changes during preschool years, the age we're working with primarily. Cognitive control has been linked with the prefrontal cortex, the cerebral cortex covering the anterior frontal lobe. So our study specifically focuses on the prefrontal cortex. We'll talk more about that later.
Okay, so now I'm going to talk about what resting state is. Recent studies have shown that the activity from neural regions that tend to correlate in response to a task also spontaneously correlate even when an individual is at rest. This is what resting state functional connectivity is: correlated neural activity in the brain in the absence of a task. Understanding resting state functional connectivity in typically developing populations is useful to help us differentiate healthy brain function from disordered brain function. Typically in adult populations, to measure resting state, they look at a crosshair for at least 5 minutes, which is very difficult to do with children due to low attention spans. However, by using new methods, it's also possible to measure this in children, and one of my teammates will go more into that later.
But why is resting state connectivity important, though? Resting state functional connectivity has been found to be associated with cognitive performance, educational attainment, and even household income. Resting state functional connectivity has been found to be associated with cognitive control in adult populations and a reliable measure of predicting cognitive control. However, very little research has been done with children, specifically in the preschool age range.
And as Elizabeth mentioned earlier, significant cognitive control changes are happening in preschool years, so we're focusing on that age range. The studies focusing on cognitive control development that do exist with children have primarily looked at activation and not resting state. To look at activation, your study needs to be task dependent. However, task parameters such as stimulus, trial duration, even computer screens vary a lot between labs, which could potentially lead to reproducibility issues. By using resting state, we avoid this issue because resting state is task independent. Studies using fNIRS, a neuroimaging technique my teammates Eileen and Christy will talk about in a few slides, have shown that the prefrontal cortex activity is developmentally correlated with cognitive control skills in children. But few studies using fNIRS have established the associations between resting state, task-free functional connectivity in the prefrontal cortex, and cognitive control development in children.
Hi, I'm Eileen Lee. I'm a senior studying art, psychology, and human-computer interaction.
Hi, I'm Christy Zhang, and I'm a senior studying business administration and human-computer interaction. Our project aims to validate an existing fMRI resting state measure using functional near-infrared spectroscopy, also known as fNIRS, with adults. We also hope to examine the patterns of resting state functional connectivity in the prefrontal cortex and the association it has with cognitive control performance for both children and adults. Lastly, we aim to identify changes in resting state functional connectivity and cognitive control performance longitudinally over time with children.
Our research contributes to existing knowledge in new ways in the following manners. Our study examines the application of NIRS for studying task-free functional connectivity in the prefrontal cortex with developmental populations, which is understudied right now because NIRS is a relatively new neuroimaging technology. Secondly, because cognitive assessment parameters vary drastically across labs, our study validates the strong association of task-free functional connectivity in the prefrontal cortex with cognitive control performance, which can be replicated in other labs, encouraging reproducibility in science, an area that is timely and important. Functional near-infrared spectroscopy is more commonly known as fNIRS, and it measures changes in blood oxygenation via imaging the brain with near-infrared light. This technique has revolutionized developmental psychology research because it is non-invasive and it doesn't require children to remain still and in a confined environment such as fMRI.
It is also less sensitive to motion artifacts like EEG. It is also relatively portable and inexpensive. Because of all of these reasons, it is very useful for neuroimaging research with children. These reasons have also contributed to its increased use in the field, which is demonstrated by the doubling of published articles in the past decade.
Hi, I'm Caitlin Cooper, a sophomore in biology, and I'm going to be talking about our fNIRS setup in the lab and about the first study that we conducted with adults. So for this study, we used a continuous wave fNIRS system with optodes mapped onto a 10 to 20 EEG cap system on the prefrontal cortex. There were 4 light sources and 8 detectors, creating 10 channels on the prefrontal cortex and 45 channel pairs, as you can see in the probe layout above. All of the fNIRS analyses were done through the publicly available pipeline, the NIRS Analyzer Toolbox. So in addition to analyzing the connection between behavior and connectivity, the study also validated an existing resting state measure known as Inscapes. Traditionally, resting state neuroimaging data is collected by instructing participants to focus on a fixation cross, as shown above, with the goal of recording their brain in the absence of performing a task. Understandably, this process is very difficult to replicate with young children who require more engaging stimuli to hold their attention. Inscapes is a short abstract film created by researchers at Yale to record resting state data in populations that can't be tested accurately by standard measures. It has been validated with fMRI in the past, but researchers have yet to confirm this result with fNIRS, so we set out to accomplish this ourselves in this study. In order to do so, we captured resting state data from all adult participants using both a traditional raster and Inscapes in a counterbalanced order, and compared their functional connectivity patterns. One advantage of Inscapes that is particularly important to note is that it is publicly available and free for all researchers to utilize, which improves replicability in future studies and accessibility in science as a whole. As you can see above, patterns of resting state from both measures were nearly identical with no significant differences between channel pairs, represented in the 2 diagrams above. Both within subjects and at a group level, this was the case. And this way, regardless of the order in which they were administered the resting state measures, this confirms that Inscapes evokes similar neural patterns in the prefrontal cortex as a traditional measure of a resting state. For a slightly different view, the scatter plot echoes the same conclusion, showing that resting state prefrontal cortex activity during the fixation cross and Inscapes were positively and significantly associated. This is a very exciting result as it opens up the field of NIRS, a neuroimaging modality that is used particularly often with children, to more accurate resting state data collection from a more diverse set of participants.
Hi, I'm Morgan Boyd, and I'm a second-year student majoring in developmental psychology and minoring in cognitive neuroscience. I'm going to talk about the behavioral assessment of cognitive control that we used in our adult study and how performance is associated with resting state prefrontal cortex connectivity that Caitlin just described. So the Stroop task is a widely used neuropsychological test that measures cognitive control. It is used frequently in screening for brain damage and performance and has also been linked to educational outcomes. In this task, color words are presented in ink that either matches the semantic meaning of the word in congruent trials or conflicts with the meaning of the word, which are referred to as incongruent trials. So if we look on the left, we're viewing a congruent trial. The word red is also written in red ink, so this task is pretty easy. But on the right, we have an incongruent trial because even though the word is green, it's written in blue ink, so blue is the correct response. During this incongruent trial, it's a bit tricky to override your knowledge of the semantic meaning of the word, so it takes a longer time to respond. So these scatter plots show negative correlations in that as mean resting state functional connectivity in the prefrontal cortex increases, mean reaction time on the Stroop decreases. Faster reaction time on Stroop performance during the incongruent trials is indicative of better performance. So these visuals show that behavioral performance on the cognitive control task is significantly associated with resting state functional connectivity using fNIRS. With children, we typically focus on accuracy as the main dependent variable because the data is less noisy than reaction time. But for behavioral tasks like Stroop with adults, researchers typically examine reaction time as the main dependent variable because adults tend to be at ceiling and reaction time data is not as noisy as it is with children. So again, these plots show that increased cognitive control is associated with increased global functional connectivity in the prefrontal cortex at rest.
Hi, I'm Marissa McAfee, and I'm a junior majoring in chemistry. I'm going to be talking about the methods and design of the second study we conducted with children examining resting state functional connectivity and cognitive control longitudinally. The participants in this study are children ages 4 to 5 recruited from Carnegie Mellon's Children's School. They are also part of the control group from a larger ongoing intervention study within the lab. The Children's School environment represents local racial and economic diversity, with children being 54% white, 24% Asian or Pacific Islander, 5% African American, 12% Middle Eastern, and 5% Hispanic. 28% of children attending also receive financial aid. The experimental protocol was approved by the IRB. Signed consent was secured from the parents of the participants, and verbal assent was secured from all participants both before and throughout the study. The children were given stickers for their participation. Our longitudinal study involved 3 major time points. At T1, the initial time point, we had 42 participants. Like most challenges in longitudinal work, there was participant attrition. 3 children moved away, so we do not have data for their 1-month and 4-month visit. 2 children's data were not collected at the 1-month visit, and 6 children's data were not collected at the 4-month visit, both due to the COVID-19 global pandemic. Other factors such as holidays, weekends, and 6-day absences also contributed to the standard deviation of the T2 and T3 testing dates. At every time point, the children completed a cognitive control task, and their data was collected with non-invasive neuroimaging.
Hello, my name is Olivia Byrne, and I'm a sophomore studying biology and double minoring in biomedical engineering and environmental and sustainability studies. The day night task shows children cards that either have a moon or a sun pictured on them, as you can see here. It is important to note that this task was developed specifically for children at this age because they can't read yet. A block of congruent trials is shown first, in which children are asked to say day when shown a sun card or night when shown a mood card. Next, a block of incongruent trials is shown where children are asked to do the opposite, say day when shown a moon card and night when shown a sun card. Each child completed 2 sets of practice trials to ensure that they understood the directions. Immediately after, they performed 2 sets of 16 test trials for a total of 36 trials. This task was also chosen because it involves multiple executive function skills such as following rules, switching, and inhibiting prepotent responses. The main outcome measure for this was the accuracy for the incongruent trials. Identical to the adult study that Caitlin explained earlier, children were also capped and shown that validated resting state measures known as Inscapes.
Hi, my name is Manasi Balusu. I'm a sophomore majoring in computational neuroscience and minoring in computer science. A one-way repeated measures ANOVA was performed with day-night task performance as the dependent variable in the time, showing the initial time point, 1-month time point, 4 months time point as the independent variables, as you can see in this graph. As you can see from this data visual, performance on day-night improves with development, adjusting for multiple comparisons, and it replicates results in prior literature that cognitive control improvements are found in preschool years, and large changes can occur in just a short period of time of a month or even a few months.
Long-range prefrontal connections are observed to increase a little over the span of 1 month and more so over the span of 4 months. The figures here show the matrices representing the strength of the connections between all 45 channel pairs within the prefrontal cortex at each time point, with warmer colors indicating stronger connectivity strengths. Connection values range from R values of 0 to 1, with higher coefficients representing stronger connectivity between channels. 1 month later, you can see there is an increased intra within brain regions connectivity, such as connectivity between channels 9 and 10, and increased inter between brain regions connectivity, such as between channels 1 and 10. You can see significant strengthening between and within areas of the prefrontal cortex as the connections become warmer with time. On the figures below, you can see each matrix projected onto a 3D brain visual. The lines represent the connections between channel pairs, and the color represents the strength of those connections. Through these, we can see increased prefrontal cortex connectivity with development. Identical to our adult study analyses, we standardized the Pearson correlation coefficients using Fisher Z transformations and averaged them together for each time point. While researchers typically focus on reaction time as the main dependent variable with adults, like for Stroop task, with children we typically focus on accuracy because accuracy data is less noisy than reaction time. In the plots for each time point, you can see each participant's mean prefrontal cortex resting state, functional connectivity on the y-axis, and their performance on day-night on the x-axis. Increased correct responses on the incongruent trials is indicative of better performance, so these visuals show that behavioral performance on the cognitive control task is significantly associated with resting state functional connectivity, replicating the pattern of findings we found with adults. Longitudinally, we found that mean functional connectivity and cognitive control performance were associated at all 3 time points, even as they both change over time.
So this is the research we've been working on for the past 3 years. We validated the use of Inscapes as a resting state measure. We implemented a within subjects design using fNIRS, where adults were presented with both Inscapes and a traditional crosshair, and found that both assessments showed similar connectivity patterns, were reliable measures of resting state, and were also both associated with cognitive control.
We also implemented a longitudinal study with children where we collected resting state functional connectivity in the prefrontal cortex using fNIRS and assessed cognitive control abilities. We found that children's performance on cognitive control tasks were positively correlated with their resting state functional connectivity changes. This study provided a unique opportunity to validate publicly available functional connectivity assessment with young children that is not dependent on a specific task parameters. And we found results that encompass the association between prefrontal cortex development and cognitive control development within the same group of children over time.
Hello again, my name is Olivia and I will also be discussing some limitations of our experiment. Firstly, we used a small sample size as a result of the limited class sizes in the children's school. This also resulted in limited sample diversity, which causes an inability to some extent to generalize the data. Sample diversity was also limited in terms of race, location, and socioeconomic status. In the future, we would like to include a greater variety of backgrounds in the children that are included in the study. Our final limitation is in regards to the duration of the experiment. Each child participated in the study for 4 months, which is significantly shorter than some long-term studies. Although following each subject for a longer period of time is ideal, this is also costly and logistically difficult. However, these are all important considerations for future directions, which Savannah will be discussing in the next slide.
Some future directions that we have include investigating specific regions of interest. For example, these areas and channels listed on the slide. We also have a lot of data that we collected that we're going to look into further, including day-night reaction time and conflict scores. Cognitive control has been shown to be crucial for later academic achievement, something we emphasized earlier in this presentation. We also collected educational outcome data, administering standardized academic achievement measures, which we also plan on delving into.
Hi, it's Manasi again. Finally, we'd like to thank the Army that made this possible, the research assistants in the Cognitive Development Lab, our advisors, Cassie Ang, Dr. Anna Fisher, Dr. Eric Thiessen, and Dr. Sharon Carver, the undergraduate offices Sura, SURF, and SURGE for the opportunity, and the Institute of Education Sciences and National Science Foundation for funding for this research. And finally, we'd like to thank the children, parents, and educators who made this project possible. Thank you.